cursory tools for actually sequencing DNA becoming available by the early 1980s, and then industrializing that process by developing new technologies throughout the 1980s, it became clear that the finite problem that was presented by the human genome with its roughly 3 billion bases was something that perhaps could be approached through some concerted effort to actually elucidate once and for all the complete sequence of the human genome. This, of course, led, after a significant amount of planning, to the launching of the Human Genome Project in October 1990, this large international consortium-based effort, highly unusual for biomedical research, with a very crystal clear goal of sequencing the human genome along with a series of other ancillary goals. As you heard in the introduction, I got involved in the Genome Project literally on day one. I was a recent MD PhD graduate, training in clinical pathology, having the opportunity during a pathology residency to actually get involved in research virtually full-time, find myself a postdoctoral fellow in Maynard Olson's lab where technologies for mapping large pieces of DNA had just been developed. I got involved at that point. We got funded as one of the first mapping centers as part of the Human Genome Project, and off I went and haven't looked back ever since. The truth of the matter is the Genome Project, while its goal was crystal clear, the path to get there was completely far from clear. It was terrifying at times. I think, as we could relay, these days of yak mapping was heroic work because in part we had no idea what we were doing. Fundamentally, we were figuring out as we went, but we had this incredible passion around the mission of the Human Genome Project. I think that passion and a significant amount of hard work of thousands of investigators around the world really led to the earlier than anticipated drafting of a sequence of the human genome published in this issue of Nature in February 2001. That was approaching the finish line of the Genome Project, but indeed the finish line of the Genome Project came when a higher quality sequence had been refined, if you will, and in April 2003, a complete sequence of the human genome was declared to have been now generated, and with that came an end to the Human Genome Project. Well, if you're counting, that was about 3,400 days ago. And a lot has happened in genomics. And in fact, the field of genomics has really sprouted up into many subdisciplines or many places where genomics has infused and exhilarated in many ways the field by enriching the scientific opportunities by using genomic approaches. Shown here are some of the areas where I would contend genomics has had a substantial influence. These are not areas I'm going to cover here. I just simply show them here to acknowledge the remarkable pace at which genomic advances influences many areas of biology and other areas of science. What I'm going to emphasize are the applications of genomics for health, disease, and medicine. Perhaps that's not surprising since I come from the National Institutes of Health in the United States. I actually have been there for 18 years. I was recruited there actually as a junior investigator in the middle of the Genome Project, rode that out, continuing to work on the Genome Project, and have been at the National Human Genome Research Institute now for 18 years. But 2.5 years ago or so, I was appointed the director of the Institute. And with that responsibility of being director of what really is the largest organization in the world dedicated to the funding of genomics research, I take very seriously the vision of what we need to accomplish as sort of one of the leaders in genomics research. Keep in mind that NHGRI was established exclusively at the beginning for accomplishing the goals of the Human Genome Project by leading the US's effort in that endeavor. But obviously, with the Genome Project completed, we've thought substantially about what we should be doing going forward. I could give you paragraph-long mission statements, but I would just summarize by saying we believe our mission now is to improve human health through genomics research. And so the idea following the Human Genome Project of applying genomics to human health and human disease was not necessarily an incredibly novel one. It was very clear that was going to be an important aspect of genomics following the Human Genome Project. The popular press started to recognize this, such as New York Times Magazine. The scientific press started to talk about this immediately after the Genome Project. And New England Journal of Medicine started writing series of articles on genomic medicine, and in fact, entire journals have now been set up, not to mention multiple textbooks that continue to come out describing genomic medicine, personalized medicine, clinical genomics, and so forth. Well, needless to say, I think a lot. In fact, that's pretty much my major part of my job, is to think about how it is that we're going to move from what we accomplished in the Genome Project to actually realizing genomic medicine. And I think of strategically what we need to accomplish in order to get us there. So let me share with you this notion of genomic medicine as I see it. It is basically healthcare tailored to the individual based on genomic information. In essence, this is going to become a clinical discipline. It's largely synonymous with what you hear referred to as personalized medicine, individualized medicine, precision medicine. I just tend to use the phraseology genomic medicine. And my view of it is that we're going to eventually get to genomic medicine, but we need to be prepared to recognize this is going to be a long journey, and this journey is one that we need to be very cognizant of how we're going to traverse. And so I think as a framework of the Human Genome Project as being the beginning of the journey. In essence, it was actually the starting line for this journey, and eventually we're going to realize genomic medicine generically defined. But we should appreciate that there's many steps that are going to be involved, and we shouldn't even think we even know what all the steps are today. Nonetheless, we can embark on this journey with reasonable confidence, having been remarkably successful at the Human Genome Project, and believing that we are going to be successful at realizing genomic medicine. And the reason we want to be successful, of course, is to fulfill the promise of why we sequenced the human genome in the first place. So what have we done as a funding agency, at least in the United States? Well, in thinking about this journey of going from the base pairs to the bedside, or if you prefer the metaphor, from helix to health, literally the day the Genome Project ended in 2003, we published a strategic vision for what should go on in genomics. And that Nature publication in 2003 actually was pretty good. And it actually provided both for our institute, but I think for the field, a blueprint of how to move forward immediately having in hand the sequence the human genome. But what has happened in the last 3,400 days, I think, has caught virtually everybody by surprise. I think the technologies have gotten better than we ever anticipated, far faster than we could have ever imagined. With that has come a tremendous amount of data, and with that has come incredible opportunities. In fact, when the Genome Project ended, I think many of us would make graphical images like this and say, someday, 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 we will, in fact, infuse genomics into medicine. But we were thinking this was going to be a decade or two out, and I think many of us are surprised that following the Genome Project, how we find ourselves even now, while not fully implementing genomic medicine, we see on the near horizon, that striking distance, opportunities that truly might come to fruition in the next few years. So as a result of that, we recognize that while the 2003 strategic plan was pretty good, the fact of the matter was time for a new one. And so we underwent another big strategic planning process, involved many people, lots of workshops, international participants, white papers, feedback, advisors, the whole set of things. And then last year, we published in Nature a new strategic plan for the field of genomics. One in particular started to embrace this concept of genomic medicine. And so this was published actually precisely in the 10th anniversary of Nature that a decade earlier had published that draft sequence of the human genome. If you're not familiar with this paper, you're interested actually in the strategic planning process that led up to it, all the workshop reports, all the white papers, all that, I'd refer you to this simple URL. You could download the strategic plan, and you could peruse all the various things that led up to that publication. What I thought I would do now is to now frame this middle part of my talk by describing the strategic plan, and in doing so, I will also be able to describe to you really what has been accomplished, that it puts us at a very unique position we find ourselves in now. What I would tell you more than anything about the strategic planning process that we conducted leading up to the strategic plan is that we heard from very smart advisors and very smart people in the community that it was time to be even more bold, more ambitious, but also more specific in describing how it was that we were going to get to the point of implementing genomics as part of medical care. And at the end of the day, one way to frame that, which is actually the framework we used for our strategic plan, was to describe research accomplishments that were going to be needed falling into 5 different domains, major domains of research activities. Let me describe for you each of these domains. So first domain, which actually was very familiar to us, was using genomics research to understand the structure of genomes, how genomes are put together. Then, of course, we're interested in using genomics to understand how genomes work, to understand the biology of genomes. We could then use knowledge of that and other things that I'll be describing shortly to you to use genomics research to understand the biology of disease, starting to get at the genomic basis of human disease. With knowledge about the genomic basis of human disease, one can imagine then doing genomics research to advance the science of medicine, how medical science actually is envisioned. We were also cautioned you can't stop there, because you can't just assume that because of a medical advance that when you throw it out into the healthcare system, either in this country or, gosh forbid, in the United States, which is a very complicated, much more complicated healthcare delivery system, that it in fact would improve the effectiveness of healthcare. So we had a responsibility as a community to also conduct research that would demonstrate that we can improve the effectiveness of healthcare through genomic approaches. So these 5 domains represent the major organizing areas for our thinking about genomics research. Now, I should immediately point out, I don't mean to imply that all science progresses from the most left side of the slide to the rightward part of the slide. and in one domain, then the next, then the next. In fact, there's many examples where discoveries in one domain immediately lead to advances in a non-adjacent domain. And many of you will also appreciate the fact that you can start at the bedside of patients and get tremendous insights about the biology of disease, and you can continue to work backwards that maybe give you insights about how genomes work and so forth. So it's not linear. These are just 5 domains with lots of nonlinear interactions among them. What's very useful for us in thinking about genomics research across these 5 domains is to consider what has happened over the last 20 years or so, and then as I'll get to at the end of my talk, what the future is going to bring. But framing it around these 5 domains and thinking about progress that you've made over different time intervals. Specifically, what do I mean by that? Well, we found it was very useful to represent, just hypothetically, I'm not going to show you this, not real data, hypothetically where the genomic accomplishments have been taking place in these domains. Let me give you an example. During the Human Genome Project, if you represent every hypothetical genomic accomplishment as a blue dot and as a density plot, as the dots all pile up on each other, they change colors, eventually becoming red. By and large, the Human Genome Project was about understanding the structure of genomes. Yeah, we learned a little bit about how genomes work, but fundamentally, the dominant parts of the activity were in that first domain. What about in the 3,400 or so days since the Genome Project? Well, I would contend that we continued to, over the past 3,400 days, to learn a tremendous amount about how various genomes are put together, and I'll describe some of that in a bit. But increasingly, we've learned about how genomes work. With that has come a little opportunities to start to explore the biology of disease and maybe even a couple of home runs out here in these clinical domains. But by and large, the center of gravity over the last decade or so has been across these first 2 domains. So what I want to describe to you now to convince you of this, but also to remind you about how far we've come in 3,400 days, is I want to go through some of these major accomplishments that create this density plot over the last 3,400 days or so. And in doing so, I'm going to continue with this theme of this journey of one step after another, getting us closer to genomic medicine. And I want to tell you about what we've done on 5 major steps. So the first step we had to deal with when the Genome Project ended in terms of accomplishments is we needed to start to understand the function of the human genome sequence, how the genome actually worked, mostly research in these first 2 domains of activity. Now, what do I mean by that? Well, let me remind you what the Human Genome Project produced. It produced this. Oh, actually, this is only 0.0001% of what it produced, but you get the idea. It was about ordering the 3 billion Gs, As, Ts, and Cs across the human genome. No pretense that we were going to understand what this code meant. It was just a foundational information for then being able to understand genome function. I'll tell you candidly that when the Genome Project ended, we didn't really know, and I still don't think we know particularly well how to fully interpret all the information embedded in the DNA code. One of the things we were good at back then was sequencing genomes, so it was very clear that one of the things we could do was generate sequences of other animals' genomes, compare them to our genome, and help to learn about what are the most highly conserved parts of the human genome, and presume that if they were highly, highly conserved, they're likely to be functionally important, so it would help give us clues about what the most functionally important parts of the human genome might be. So as the recognition that humans being just one little insignificant twig on the phylogenetic tree of vertebrates, for example, it was time to start to explore these other branches. We started out first with some obvious animals like mice and rats, also companion animals like dogs, but also some of our closest relatives like chimpanzees. But we also recognized that if we really wanted to be robust in finding the most highly conserved parts of the human genome, we needed to sample across multiple different phylogenetic branches of the tree, and that's why various critters, including some Australian critters, were selected for genome sequencing. About 36 or so mammalian species have now had their genome sequenced. If I was pointing to one paper, was this one that came out, I think to date is the most comprehensive analysis published last year across 29 mammals that are now pointing to some of the most highly conserved parts of the human genome that are conserved virtually across all mammals. What have such comparative genomic studies and some of the earliest other studies annotating the human genome have revealed? Well, it's revealed, for example, that across the 3 billion letters, about 5% of our genome sequence is under high evolutionary selection, selected, in other words, conserved across virtually all mammals. But what's been surprising is out of that 5%, only about a third of that 5%, about 1.7%, directly codes for protein coding genes. This accounts for about 20,000 genes. We have a pretty good inventory of those genes now. But the fact of the matter is we have now come to learn that about 2/3 of the most highly conserved part of our genome we need to highlight in a different color because it is clearly as conserved as coding regions and almost for certain biologically important, but is functioning in ways other than directly coding for proteins. These are regulatory sequences, small RNAs, various other ways that we now can begin to appreciate that DNA can confer function. And of course, we've also learned that it's not just about the primary DNA sequence. The last 3,400 days, we have greatly become to appreciate that the epigenomic landscape is critically important for human biology and human disease. It's these methyl groups that decorate our DNA. It's these histone marks that are critically important. And increasingly, we need to be inventorying all the epigenomic aspects of the human genome as well. That is the reason why NHGRI launched an effort a number of years ago called ENCODE. It's another one of these international consortiums. ENCODE stands for Encyclopedia of DNA Elements, which is aiming to catalog lots and lots of information about the functional landscape of the human genome, both using computational methods as well as high-throughput experimental methods. Increasingly, any of you can go, all the data, of course, is released, like all major genomics projects. You can go to various genome browsers and you could pull up different regions. Shown here are 2 regions, and you will be overwhelmed with ENCODE data. I can assure you of that. With information by these various horizontal tracks showing about conserved regions and gene models and which DNA is being made into RNA and which bits of DNA are being bound by different DNA binding proteins and so forth. And in fact, it becomes almost an overwhelming amount of data. So the consortium last year published a user's guide, if you will, to help individuals sift through that data in an attempt to try to understand exactly what the functional elements are in a particular stretch of a human genome they're interested in studying. ENCODE continues to march along. I can tell you that next month, in very early September, there will be a major publication in a very prominent international journal describing ENCODE's latest accomplishments. That will be accompanied by about 3 dozen companion papers that will be appearing in 2 other genomics journals as well. So stay tuned. ENCODE will continue to march along, and with it will come better and better insights about all the functional aspects of the human genome. That said, decades from now, we'll still be interpreting the human genome sequence. There's no reason to think we even know all the ways DNA confers function. There's no way to think even now we have a complete inventory of all the functional elements that we need to understand. So that was the first step. The second step really relates to the fact that we're not just interested in how a hypothetical human genome functions. We're interested in how each of our genomes function and how our patients' genomes function. The reason, of course, we're interested in that is because every one of us has a slightly different genome, because we vary. So there's a second step along the way is to understand human genomic variation, activities that have mostly been focused around these first 2 domains of research activities. Now, specifically, what do I mean by this? Let me remind you, each of you consists of about 6 billion base pairs. You got 3 billion from mom, you got 3 billion from dad. If you look to the person sitting next to you on your left or your right, at a single nucleotide level, you differ from that person across your 6 billion bases. There's about 3 to 5 million places that you have a different nucleotide, that you vary. So these multiple variants, 3 to 5 million that each of you probably has. Now, it turns out, of course, that the great majority of these variants have no phenotypic consequence. They're biologically silent, if you will, but a subset of them are relevant. A subset of those might confer risk for a disease, might be detrimental. Another subset of them might be good variants to have. They might be protective to a disease or be associated with some other positive attribute. But the other thing about your 3 to 5 million variants is you don't have your own private 3 to 5 million variants. Some of them might be pretty rare, just you and your family, but most of them are probably pretty common. In fact, we probably share across this auditorium many variants in common, and across the world population, many variants. Most of the variants, in fact, are going to be far more common than private in your particular family. So that becomes a finite problem, and anytime genomicists see a finite problem, we tackle it with both feet. And the fact is, it was recognized that wouldn't it be wonderful to start developing catalogs of at least the most common variants across the whole world from different world populations. That started with something called the SNP Consortium for Single Nucleotide Polymorphism Consortium, but then got far more organized around another international consortium called the International HapMap Project. And through 3 successive publications, this international effort described lots and cataloged and made available information about millions and millions and millions of variants, especially the more common ones existing across different human populations. In addition to that, they characterized information about how those variants relate to one another when they sit on chromosomes nearby in structures known as haplotype blocks. And haplotype blocks are basically just stretches of DNA across on your chromosome whereby the variants that sit near one another tend to segregate together as a block, as a neighborhood from one generation to the next. They tend not to have recombination happening. And so as a result, that architecture is actually was very important to know what it might look like. And the HapMap project basically elucidated that architecture in a way that becomes very useful for human genetic studies, and I'm going to tell you about in a couple of slides. When better technologies became available, we got more ambitious. A new consortium was launched called the 1,000 Genomes Project, which aimed now to use sequencing to try to get at rare and rare variants, again, across carefully selected individuals from different human populations. It's an underachieving consortium, it's an overachieving consortium in that, sorry, that was a slip, is that it's not just 1,000 genomes, it's now studying over 2,400 genomes. A pilot project aspect of it was published late in 2010, once again, in an international journal, prominent, you will be reading later this year, a major publication about the first major phase beyond the pilot of the 1,000 Genomes Project, and I think you'll be impressed with how much information is now available in databases about genetic variation. But that, of course, just sets up the third step along the way. The third step along the way, of course, is using that information about genetic variation to try to get at the genomic basis of disease. And here we now are moving into this middle domain of activity. And of course, this now starts to approach the notion of being able to correlate variants with phenotype, in particular disease phenotypes, which we're also interested in. Now, in order to describe what's happened in the last 3,400 days, let me remind you a couple of things about human disease. Virtually all human diseases have a genetic influence or an overt genetic cause. And you could broadly divide human diseases into two general categories. Let me tell you about each, because what's happened for each over the last 9 years is slightly different. On the one hand, you have rare diseases, rare genetic diseases such as sickle cell, cystic fibrosis, Huntington's disease, and so forth, whereby they're genetically simple because it is defects in a single gene that represent the major risk for getting that disease. Yeah, there might be other genetic variants elsewhere in the genome that influence the severity of disease, and there might even be some environmental contribution. But by and large, you're looking for the defect in a single gene. These are known as monogenic disorders, also known as Mendelian disorders, named after the famous geneticist Mendel. Well, rare genetic diseases are devastating to families and to patients, and they're incredibly important and insightful to study. But the fact of the matter is they don't represent the major healthcare burdens around the world. What fills hospitals and clinics around the world are these diseases. It's common diseases. The thing about common diseases is that they're genetically complex because they're caused by multiple variants that exist in the genome that all conspire together with what is typically a greater influence of the environment to give some greater likelihood of getting that particular disease. So these are also known as multigenic disorders or non-Mendelian disorders. Well, the idea, of course, of sequencing the human genome would be to accelerate the pace at which we could characterize these various variants, ones that overtly cause disease and one that influence or confer risk for getting disease. And there was always great optimism that the Human Genome Project would accelerate the elucidation of genes associated or responsible for rare diseases. And there was real question marks about what was going to happen when you actually tried to apply strategies to elucidate the genetic basis of complex diseases. Let me tell you what's happened in terms of advances in each of these areas. So starting with rare genetic diseases, shown here is simply a cumulative plot of genes that have been identified that's shown to be, when defective, causative of rare genetic diseases. These are single gene disorders. Let me remind you, the Human Genome Project began there. With the day the Genome Project began, there were about 36 genes that had been implicated in rare genetic diseases. And it's hard to argue that as soon as some of the earliest maps and sequence of the human genome became available, it accelerated the pace at which we could discover the genetic basis of rare diseases, and that's continued up to the present time. In fact, it's truly remarkable because where once we had just 36 genes that we had identified, we now have about 3,500 genes. In fact, nowadays you open up journals all the time, continually finding more and more cases where you've identified the gene for a rare genetic disorder. That is the glass half full. There is a glass half empty component of this pie chart because you can see there still remain about another 2,000 or so Mendelian diseases and traits for which we don't yet know the genetic basis, and another 2,000 or so on top of that we think there's a single gene involved, but we don't yet know the genetic basis. So great progress, but nonetheless, great challenges ahead. Memorize this pie chart. You're going to see it later in the talk. What about complex genetic diseases? Well, for complex genetic diseases, we always knew this was going to be difficult, and people had various ideas on how to tackle the problem. One of the ideas that really has come to the fore over the last 5 years takes advantage of knowing this architectural structure of human genetic variation with respect to these haplotype blocks. So for those of you, again, who are not human geneticists, who aren't familiar with genome-wide association studies, let me digress and just explain these to you so you can appreciate what's happened. Shown here is a stretch of a human chromosome, and along the top, each of these little black lines represents the position where there is a single nucleotide variant, a single nucleotide polymorphism. Your inverted red triangles represent these haplotype blocks. So all these genetic variants on this inverted red triangle tend to hang together as a neighborhood from one generation to the next. And the same for this triangle, the same for that triangle, and so forth. Well, the idea was you want to figure out, by detective work, if you will, whether or not specific regions of the human genome with specific variants tend to be inherited with individuals with a disease compared to those without a disease. So what do I mean by that? The study is designed, you take 1,000 people with hypertension and 1,000 people without hypertension. You ask the question, are there any parts of the human genome and any variants that tend to occur more often in the hypertensives compared to the non-hypertensives? Problem is, we don't want to start assaying individuals for millions of variants because it gets too expensive. But the idea was, well, wait a second, we don't need to assay all the variants here. We can just take 1 or 2 of the variants in this particular block, and it will be a proxy for all the other variants in that block. So how does that play out? Well, for example, let's say we took a variant from that block, and let's say the variant, for simplicity reasons, just comes in a green flavor and a purple flavor, and these are your hypertensives and these are your not your hypertensives, and you can sort of see that there's no real correlation. There's no statistical association. But if we go over here to this block, now we have an orange variant and a blue variant, and wow, it seems like the orange variant tends to be associated more with the hypertensives, whereas the blue tends to be more associated with the non-hypertensives. There is an association. Well, you could do this by block by block by block by block across the whole genome, make this genome wide, and evaluate whether or not you see a statistical association. You would rule out a block like this is not in the hunt, and you would rule in a block like this that requires further study. Now, let me point out, it's not that the orange variant is causing or conferring the risk for hypertension. It could be one of the other variants hanging out here, but at least you've reduced your space down to one neighborhood. Now you can interrogate this block much greater, find out what all the variants are, and then figure out which of the ones it's actually conferring risk. When you do that, you do it genome-wide. You can collect data and get plots like this, where now you've just surveyed across all the blocks across the human genome. And here's, for an example, a block where there's high statistical association with this particular marker on that particular block, and that you would follow this up. Well, the interesting thing about genome-wide association studies is that in about a half hour, you can make a beautiful PowerPoint slide to explain it. And the problem was nobody had any idea if it was going to work or not. It was all a back-of-the-envelope calculation that we should be able to get enough statistical power. So it was very reassuring that when some of the earliest HapMap data came out, even before the full HapMap project was even completed, by 2005, there was a success story. The first successful genome-wide association study, in this case for age-related macular degeneration. And they actually got fortunate in this case, they could figure out the gene. Most of the cases, you don't know the gene when you get statistical association. Well, at NHGRI, we decided this was going to be an important area to track, so we actually developed a system for tracking the literature and monitoring this and developing lots of resource information. And just shown here graphically, in 2005, right there is the region of chromosome 1 that was found to be statistically associated with age-related macular degeneration. So we stuck a little lollipop there to indicate. By 2006, there were a couple of other successful genome-wide association studies. By 2007, it almost got to be the point that every single time you opened Nature or Science or PNAS or Nature Genetics or PLOS Genetics or any of these major journals, you would see another successful genome-wide association study. And that trend really continued, and we just kept putting down lollipops wherever the regions were that were implicated. The trend continued throughout 2010, and in fact, the trend continues up into the present time. This is the most updated graphic. Once upon a time, 2005, 1 successful genome-wide association study. As of today, 1,200 successful and published genome-wide association studies. We've littered the human genome with lollipops showing associations for over 1,000 regions for about 250 different human diseases, all the diseases that you might readily name as being clinically important. And we now have not necessarily genes, and in some cases probably not even things that have to be followed up, but at least we have regions of the genome by which we can now study more aggressively to figure out what might be the underlying variant that is conferring risk for having that disease. So this, once again, is sort of a glass half full kind of a thing. We've really advanced human genetics research, but obviously have lots more work to do. Among the hard work we have to do ahead of us is a realization that's come out of these studies. Because what we now know 3,400 days after the Genome Project ended and all these studies about human genetic variation and these genome-wide association studies and so forth, and advances in rare genetic disease research, is that the great majority of rare diseases are turning out to be caused by mutations that sit in coding regions, protein coding regions, the part of the genome we really understand, so it's a straightforward thing to figure out and show mechanistically what's going on. The glass half empty is the fact that we are now learning that these genome-wide association studies in the majority of times seem to be pointing to non-coding parts of the human genome, that the variants that are conferring risk for these incredibly important clinical disorders are probably residing in the parts of the genome that we don't understand as well, the non-coding parts of the genome. It should be very motivating to figure out how those work, but it also means that characterizing these and showing causality is going to be quite challenging. Nonetheless, it's going to be very important. One of the things we now know as of today that we clearly are going to have to be able to do to take these associations to the next levels, we need complete information about it of all variants sitting in any of these associated haplotype blocks. And we need this across thousands of individuals and thousands of controls. And in order to do that, it's very clear that the next step of human genetic research for complex genetic disease is going to have to involve the ability to routinely sequence whole genomes. Now, this fourth step, as I'm sure many of you are familiar with, is probably the most successful thing I'm going to tell you about in this talk today. It is truly remarkable what has happened in terms of sequencing technologies. It's influenced so far mostly these first three domains, but needless to say, I think as we go in the future, you're going to see these sequencing technologies having great influence on these other two clinical domains as well. Well, what's the origin of this? I must admit, I think it is worth pointing out that this just didn't happen overnight by magic. In fact, in 2003, when we published the strategic vision I introduced you to earlier, I was a co-author on this with other leaders at the Institute. And we said a lot of really audacious things because it was the thing to do when the Genome Project ended. But one of the more audacious things we said was that we needed technological leaps that seemed so far off as to be almost fictional, but which, if they could be achieved, would revolutionize biomedical research and clinical practice. Well, that sounds pretty fluffy. But then we actually gave a specific example. Because we said, for example, we need the ability to sequence DNA at costs that are lowered by 4 to 5 orders of magnitude than the current cost, allowing a whole human genome to be sequenced for $1,000 or less. Now, why was that such an audacious thing to put into press at Nature? Because this came out the day the Human Genome Project ended. We had just sequenced the first human genome, and it cost $1 billion, roughly. And here we were proposing that we want to get to the point where we have developed technologies that reduce the cost of sequencing to something like $1,000. This gave rise to this battle cry of the $1,000 genome. We, among other funding agencies, funded a whole bunch of groups to develop new technologies, crazy ideas, reasonably crazy ideas, not so crazy ideas. We wanted all ideas. And the good news is we were met by the private sector putting a lot of venture capital money in and a lot of companies sprouting up. And all I could say is I've been involved in genomics a long time, and we always had a vision of getting rid of a factory like this. That was the one of many that produced the first sequence of the human genome. And we had an idea of having some fancy technology, a micro this, a nano whatever, that would be able to sequence DNA incredibly inexpensively. But I don't think any of us anticipated it happening as fast as it's happening and how it's really just catapulted the field forward. And it's not that we have one technology or two or three or four or five or six or seven. It's that we have like 8 and 9 technologies that are now available. Just shown here are some of the commercially available instruments. But don't worry about it. Don't memorize this slide. In 6 months, I'll have to update it, and a year later, I'll update it again. There are more technologies coming along, and these next generation platforms are just truly remarkable with respect to what they can do. And they've just been game-changing, not only for human genomics, it's been game-changing in many areas. You may ask, where are we in getting to the $1,000 genome? I can tell you very precisely, because as of today, we are right there. Okay, we're really not quite at $1,000 yet. And many of you might have heard you can do a shortcut. You can just sequence the coding regions of the human genome or just the exomes. You'll hear about whole exome sequencing. That's less than $1,000. Many companies around the world now offer whole exome sequencing of the human genome for $800, $900, even less in some cases. But we will absolutely get to a whole genome sequence for $1,000. And this has been remarkable. Now, let me give you some real numbers. And it's not just how cheap the sequencing is, it's also how quickly you can get that sequence. So as I told you, the Human Genome Project, when we were actually sequencing DNA, the first real sequencing of the human genome, it took place over about 6 to 8 years of sequencing activities, and it cost $1 billion. Now, the day the Genome Project ended, we actually calculated what it might take to sequence a second human genome. And when we did that back of the envelope calculation, we thought, well, if the same groups just started sequencing again, it would take 3 to 4 months, but it would still cost $10 to $50 million. Today, what does it cost? Well, today, it takes about 2 to 3 days, and it's something on the order of $4,000 to $8,000. We already know one instrument by the end of the year, it'll be down to 1 day, and we also can anticipate probably by the end of the year, it'll be down to $3,000 to $4,000. I don't worry about this anymore. With my eyes closed, I know we will coast to a $1,000 genome. I know that because of what's already been available. I also know that because stuff that's being published. For example, some of you might have noticed Nature Biotechnology published a special issue where they started talking about nanopores. That's the new thing in sequencing. Oxford Biosciences has announced now that they have methodologies that they're going to commercialize in 2 instruments where these nanopore molecules sit in these lipid bilayers and DNA strands just get pulled through and they can just tick off exactly what nucleotides are there. It's just incredible technology. I have no idea if this company will be successful. I'm not endorsing anything. One thing I do know is that in February, they announced that 2 instruments they're going to commercialize later this year. One is a desktop instrument. Their other instrument, which will sell for like $800 or $900, is a USB device that plugs into your laptop, and will sequence DNA, claiming a human genome in something like a day or 2. I just think that's like way cool. And what's even better than that is that this USB device will work in a Macintosh or a PC. So they have the right idea. So bottom line is, I'm not worried about this, and I don't even think this is the last technology. Sequencing technologies are going to get better and better, and we find ourselves suddenly in a position where data generation is not rate limiting, that the cost of generating the data has gotten almost to the point of being trivial. That's the glass half full, but you're getting used to me at this point. I'm going to tell you about a glass half empty, because it's the 5th step is the thing that keeps me up at night. And that's the ability to actually analyze all of this data. Simply put, we are the victims of our own success. And this is a problem that is absolutely going to hinder activities in all of these research domains. It's not limited to any one of them. And by being victims of our own success, I mean that we've created a new bottleneck. It used to be data generation, but now the bottleneck is data analysis. Because we have these new technologies, and they spew out sequencing data far faster than we can assimilate it, far faster than we can even deal with it. And it's put biomedical researchers in a place that they've never been before. I remember when the Genome project was going, we used to say, we're generating a lot of data here. You talk to a computer scientist, and they'd laugh. They'd say, you hardly have any data. Particle physicists, they have data. Astronomers, they got big data. You don't got big data until now. And I think everybody's realizing this, including the scientific literature. We have big data for the first time. And it's caught us in some ways by surprise because we didn't think these technologies would move us so fast. But they've put us here. And there's aspects of this that you need to dissect. There's sort of computational problem. There's also informational aspect of the bottleneck. On the computational side of it, I mean, it's some of the obvious things, just having enough servers to store all the sequence data, having enough processors to analyze it, even having bandwidth to push it from site to site ends up being a problem. Software. Each of these technologies are imperfect, and you need software tools that allow you to analyze the data and deal with all of its inaccuracies and putting it all together and detecting variants and so forth. And we don't just need software tools for the big genome centers. We need software tools for every one of you and all investigators around the world. And those software tools aren't yet available. We need to be developing them. Oh, and for all the trainees in the audience, please pay attention. Workforce. We really need to be thinking about what the next generation of scientists and physician scientists look like. And the notion of being able to have the next generation prepared for the quantitative science that biology is now becoming with all this big data is something that many of us, including those of us at NIH, are thinking about training the next generation and thinking about how to alter the training programs to make sure they're well prepared. It's too late for those folks like me, but for the next generation, wow, we should be thinking about what that world's going to look like. Well, we'll solve this computational bottleneck, but we shouldn't believe that that's the only problem in town. Because the fact of the matter is, even on any given day, we can use these fancy schmancy sequencing technologies of getting a genome sequence of any human, any clinical subject, and we can even slog through the big data issue and even generate our list of 3 to 5 million variants that any one of us or any one of our clinical subjects might have. But trust me, when we go variant by variant by variant, for the great majority of those variants, we stare at it and we have no idea if it's an important variant or not. And the truth is, we are already sequencing many human subjects as part of clinical research and sequencing their genomes. But let's be real and realistic. When we go to round on those patients in the morning, we look at the variants and we're left wondering what most of these mean. we have a major need for being able to move now forward to better understand what these variants mean and having them in a form that's going to be very useful for scientists, for clinicians, and so forth. It's actually a reason why Harold Varmus, who Nobel laureate, previous director of NIH, now head of the National Cancer Institute in the United States, he wrote in a commemorative editorial a couple years ago that physicians are still a long way from submitting their patients' full genomes for sequencing, not because the price is high, because it's not high anymore, but because the data are difficult to interpret. And then good friend, good member of the community, Lane Martis, humorously wrote recently, talking about the $1,000 genome, but the $100,000 analysis. And while it's sort of funny, it's sort of not funny at the same time. We've got to fix this, and we will, and we're thinking of ways, but it really represents, realistically, a big challenge that we are facing now and will continue to in the future. So those are the 5 steps that I wanted to tell you about that I think sort of frame what's gone on the last 3,400 days. But obviously, we're not at genomic medicine yet. These new clinical-oriented domains are ones that are really coming into focus and ones we are certainly thinking about, and I'm sure many of you are thinking about, whether it relates to developing new diagnostics, using genomic information to develop new therapeutics, obviously using genomic information perhaps to develop better preventative strategies, and of course there's unknowns and unknowns and unknowns that we still need to face and we will face. So with that as sort of the framing of the present tense, I just want to spend the last 10 minutes or so as promised and drag you into the future a little bit. I'm sure many of you are interested in hearing what I predict might be in store for us in the future. And in starting that, I want to continue on this density plot and fill in the rest because I think it'll frame what I want to say very nicely. What's going to happen the next decade, for example? Do I sort of see things just instantly moving where we're going to be changing the face of medicine? No, not at all. I think we have to be realistic here. Changing medical care and medical practice is extremely hard. Even making medical science advances is extremely hard. That said, I think I've convinced you, I hope I've convinced you that we have really teed up opportunities, in particular for getting at the biology of disease over the next decade. I think the future, hypothetically, is going to look something like this. I think the next decade, more than anything, is going to be about getting yet a more sophisticated view about how genomes work. But I think the other highlight of the next decade is going to absolutely be getting a better understanding of the biology of disease. I think with that will come more and more opportunities for advancing medical science. And yeah, maybe we'll even see more blue dots out here where we really have changed the effectiveness of healthcare. Realistically, though, I don't think we're going to change the face of it in the next 10 years. I think it's coming. And I think beyond 2020, we should be all optimistic that the center of gravity will continue to march along to the right. But realistically, I think it's not going to happen over the next 5 years and not even sure over the next 10 years. What is this going to look like, especially with respect to advancing our understanding about the biology of disease? Well, I will tell you it will involve sequencing a lot of people's genomes. And it's not just the people shown on this slide. Those are only thousands. It's not even these. It's tens of thousands. No, this is more like hundreds of thousands. I think that's what we're looking at. I can tell you at NIH, we recently did an inventory of the grants across NIH that have been given out that are going to involve genome sequencing. And we figured out that over the next 3 years, NIH only will be generating upwards of 80,000 human genome sequences as part of their research. And that's just one funding agency, one country. I think worldwide, we're talking about hundreds of thousands of individuals over the next few years alone. And I'm way beyond that when you sort of even get over the next 3 to 5 years. Now, this is not going to be part of clinical care. I think these genome sequences are going to be part of clinical research predominantly, such as at major government and academic research centers. Increasingly, it will involve physicians thinking about how to use this information. But again, I think this is for the near term going to be around clinical research endeavors because there's so much that needs to be done research-wise to inform what's going to happen. But that's a lot of genomes, hundreds of thousands of genome sequences, and they will pour into the databases. And increasingly, I can tell you at NIH, we are trying our best to figure out ways not just to have them go here and there and wherever, but the power of having that much sequence data aggregated in one place, along with lots of information about clinical phenotypes and the metadata associated with that sequence, makes it all more powerful, and of course make this all available so that anybody around the world, scientists, can then take advantage of all the sequence data and all the associated other data to be able to compute in their ways and come up with creative ideas for being able to analyze that data. So the goal very much is to get this data as it's coming in an organized way to allow lots of people to analyze it in all different ways. Well, what's it going to look like in terms of specifics? Can I give you specific projects? Well, let me tell you about what we're doing at NHGRI, several examples of what that future looks like, because these are things either we just started funding or that we're going to start funding over the next year or two. A lot of them are going to involve these fancy sequencing technologies. And for example, I told you to remember this pie chart. We just started a new program where we've set up several new sequencing centers who are going to focus on getting at the genetic basis of the remaining Mendelian disorders. And doing so in a very community-based intake fashion, where around the world they will collect DNA samples from patients whereby they probably in many cases are sufficiently rare that family studies have not yielded genetic clues or other approaches have not yielded genetic clues. They will industrialize the process of sequencing, interpreting, and trying to figure out which the causative variants are in those individuals. We hope to fill in much of this pie chart over the next 5 or 10 years. Meanwhile, we're very cognizant of the notion that we need to accelerate and figure out how to move forward on going from these lollipop regions to actually showing which variants are the risk-conferring variants. So some of our largest sequencing groups in the United States in particular are going to be focusing around complex genetic diseases. We will be doing a major project in Alzheimer's disease, for example, similar ones in various other complexes where large numbers of samples are going to have to be sequenced to get the kind of statistical power to be able to go to the next step for complex genetics disease research. A common question I am asked is, what is some of the earliest wins that for clinical applications in particular? In other words, what is some of the lowest hanging fruit out there? I'll give you 2 examples of what I think some of the lowest hanging fruit. One I think is becoming increasingly obvious, and that's around cancer genomics. Cancer is a disease of the genome, and the fact of the matter is we can use these powerful sequencing approaches to now inventory the aberrations that take place in cancer specimens. In the United States, the Cancer Genome Atlas is our big project. It's a joint venture between us and the National Cancer Institute. But this is all part of a larger and incredibly productive international consortium of groups, including groups here in Australia, whereby many different kinds of cancer, the consortium is called the International Cancer Genome Consortium. Many different types of cancer are being targeted for detailed study, hundreds of specimens collected, lots of sequencing done, inventories of common aberrations, and we are learning a tremendous amount about the genetic and genomic underpinnings of cancer. With that, it's going to come great opportunities in diagnostics and stratification, and hopefully will also come some insights about new therapeutic strategies. But I would say absolutely, the changing in clinical practice is going to come first, I think, in oncology, probably before many other disciplines. Another low-hanging fruit related to these fancy technologies and lots of sequencing that's going on will be in the pharmacogenomics arena. Drug response clearly has a genetic basis, and it's a complex trait in many cases, but these genome-wide association studies, like many other studies, are revealing the genetic basis of drug response, and already there's multiple medications for which it is clear that having genomic information prior to decisions about what medication to give a patient is standard of care, and I think those numbers are going to grow into the dozens in a matter of just a few years. Well, those are some of the low-hanging fruit, but we're not just tackling the easy things. We're prepared to tackle the hard things, and I think there are some really hard problems ahead of us. Some of those hard problems are sort of summarized by, enough with the low-hanging fruit. How about some slow-moving meat? So we are tackling some meaty problems, too. Let me give you examples. I've talked to many people this week about this because it's always interesting to come to another country and hear thoughts about newborn screening. This immediately elicits lots of interesting ideas and ethical dilemmas and so forth, and every country does it a little bit different, I think views it a little bit different. I've come to learn that Australia, like America, has probably virtually its entire citizenship. Its first encounter with genetics happens at about a day or 2 old. Because you have mandatory newborn screening where a small amount of blood is taken from a child's heel, put on a little filter paper, and off they go and they get genetic testing for, I understand here, several dozen genetic disorders, similar numbers in the United States. And one could imagine that the number of disorders that one might want to screen for will grow with time. And one might imagine that at some point it's going to be simply more efficient to sequence that DNA rather than test it for some battery of genetic mutations. But wow, that brings up many issues. What do you do with all that information? Do people want to know, et cetera, et cetera, et cetera? What we are doing to try to think about this future is to research that future. And so our institute is now partnered with the Child Health Institute, and we've just have out now a call for applications to explore what the future might look like with these new sequencing capabilities in the arena of newborn screening. And of course, that will include studies of some of the ethical and social implications of this kind of research. We believe we need data to support sort of thinking about how to move forward in this area, and we think data would be very much generated by the kinds of studies we hope to support. We also realize a meaty problem is disseminating information about genomics. We fully recognize that there's a tremendous amount of scientific accomplishment that's taking place, and that information is overwhelming physicians who want to simply figure out what to do with their patients, but are sort of overwhelmed by the amount of genomic knowledge that is burgeoning every day and every week. Meanwhile, physicians are going to be increasingly asked to communicate to their patients about genomics, and that's complicated around the fact that patients are going to struggle with having some minimal genomic competency and literacy, and we need to think about what that looks like. So we're tackling that. For the former one, in particular, we have heard loud and clear that we need better and better information systems to deal with clinical genomics. And as a result, we've now launched some initiatives to sort of pursue this. This very much ties into the electronic health records, which I know this country is dealing with, and certainly the United States is dealing with, and genomics just feeds seemingly nicely into an electronic environment, but there's many challenges that have to be dealt with along the way. But increasingly, we also know we need to have well-curated information available to practicing healthcare professionals who can be a couple clicks away on an iPad to find out if a given variant, what it means. They don't have time to go look at the primary literature. We need to curate this and make this available. Otherwise, this is simply not going to be acceptable to the healthcare profession. So again, we're launching some initiatives to start to research and pilot what this might look like. And finally, of course, we recognize in the United States, like Australia, has been highly cognizant that there are many societal issues that have to be grappled with. For many years, we've dedicated 5% of our research, it's actually required, to studying the ethical, legal, and social implications of genomics research. And we continue to do this and think about many, many issues that become very relevant, especially as you start to get more and more relevant to people as part of their healthcare. And this will be something we certainly will continue in the future. So needless to say, and in wrapping up, it's very clear that sequencing is already starting to alter the clinical landscape in ways that we can anticipate, but also ways that we need to study to better anticipate. I will tell you, and really the second to last slide, is that what also becomes very, very difficult and challenging for us is the recognition that as we're moving along from the left side to the right side of the slide, we get out of our comfort zone because of the nature of who genomicists are. Most of us, like myself, who sort of grew up in the Genome Project, consider ourselves basic scientists. And understanding how genomes are put together and how they work and how they influence disease is mostly a basic science endeavor. Increasingly over the last few years, as we've gotten into looking at the more sophisticated studies of human disease, it becomes translational science, which of course takes us into the domain of looking at medical science. But the fact of the matter is, to really change healthcare, it's not even just about translational science, actually a new science that I'm beginning to learn about called implementation science, where you really have to practically study aspects of healthcare delivery and uptake of healthcare information and so forth. And I admit, I think we as a community, certainly our institute and me personally, we're students of this, but increasingly we're trying to attract individuals who have expertise. And we recognize that we need expertise to help us make this full progression. So it's just one of many challenges I think I've introduced you to today. So let me close, and I'll close with a quote. And the quote was a quote I came across a number of months ago, Winston Churchill, where he said that a pessimist sees the difficulty in every opportunity. I hope I've convinced you that genomicists are not pessimists. We love opportunities, and we don't even mind difficulties. We're optimists. And when we see difficulties, we see great opportunities. And I think the strategic plan that I've sort of alluded to throughout my talk and the future that I try to describe to you is very much about seeing those difficulties, but recognizing them as opportunities because of the compelling nature of what we're trying to accomplish. So I will stop there, and I'm happy to take any questions. Thank you.
Questions for Eric?
Right here.
I've got a question that relates to the multigenic diseases where you expect a relatively large number of genetic variants with minor effects.
Yeah.
So when you have that situation, each of these variants will produce maybe a difference in the order of 10% or less to some biological function. And we know from allelic series in mice that a difference in 10% of RNA level, even less protein level, can have cause a phenotypic difference. But it's very difficult in such experiments to reach statistical significance of your experimental system, which limits publication. And likewise, well, but it makes
a second part question, or is it
no, this is all the same part.
Okay.
So that makes it difficult to publish. But likewise, if you had a multi-genetic variant disorder and you wanted to and you had or needed to target all of the effects with different drugs, and then a combination, say, of 10 drugs would treat the disease, each of these drugs individually would have a minor effect, which would be difficult to develop for the clinics because
so if I understand the nature of the question, I think I do. I don't think anybody is claiming that we're going to get to a specific variant, a little variant here that gives a little bit of a tweak that results in giving you a little bit of risk for hypertension, and that we're going to develop a drug to that variant. Rather, the path that is being described is one where we get at a variant that tells us that there's this gene involved or a regulation of a gene involved, and that puts us in a pathway. And then it's the pathway that then gives new insights that where pathway has now been implicated, for which you figure out through other means ways of tweaking that pathway. And in fact, what we've learned from some of the early GWAS successes is that some of the drugs that have been used for years are turning out to be hitting pathways that are being hit by GWAS, those genes are being implicated. So I don't think it's variant by variant, even gene by gene. I think the paradigm, although it still has to be proven, is that you use this to figure out what pathways need to be tweaked, and then use pharmacological interventions to tweak the pathways.
Given that we've moved from a small data science to a big data science, one of the big problems is that all our training and the regulatory systems like journal reviewing and grant reviewing have all struggled to keep pace with the move into more quantitative sciences. There's also going to be a parallel to that once we move into healthcare as well, that there's going to be this big lack of understanding of the quantitative issues and the real scale. How do you think we should address that?
One issue at a time. You can sort of deal with each of you. Deal with regulatory issues, journals. Let's take the training, because it's one that we're dealing with very aggressive, or we hope to do it aggressively at NIH. I think you have to look to say, are we training biomedical, scientists of the future in an appropriate way to deal with this new era where quantitative sciences, whether it's computational biology, bioinformatics, biostatistics, medical informatics, they have to be facile at these things. And so you can mandate some minimal competencies in those areas. So I think we should be thinking about that. And maybe we'll all of a sudden attract people that previously thought biology wasn't for mathematicians. Well, it probably is. And so I think we need to see a shifting of the people we attract and or what we do with the folks that come into our training programs. Journals, you've got to deal with, these are all policy issues. There's a lot of cultural change we need to have. I think genomics, some people joke that, or no, they don't joke, I think they claim that the Genome Project produced two things. It produced sort of the sequence of the human genome and a complete cultural shift about how biomedical research is done with respect to data sharing and so forth.
Thank you very much for a really interesting lecture. You mentioned several times about the importance of collaboration and the large scale. And I was wondering if you wouldn't mind making some comments about the digital divide and who's getting left behind and the kind of unevenness of the distribution of the skills and the technology and what this means for sharing and as we move towards your fourth.
And do you want me to do you want me to answer this with respect to patients, the divide, or are you talking about among the scientists?
No, I'm thinking more about, I guess it gets delivered to patients.
But you're talking about scientists.
And countries.
Yeah, country. So what I very much appreciate your question, and as I can tell you, we're just in the process of launching a major genomics initiative in Africa, and jointly with the Wellcome Trust. To try to infuse genomics into the scientific enterprise in Africa. So I'm very appreciative of some of these issues. What I would say is I think genomics gets a lot of credit for this because, being fortunate that it's electronic, this notion of open access of data, that the cultural shift where data is made available, put up on the internet for anybody to access, I think has really been a change in how biomedical researchers have previously operated. Because all you need is an internet. You need an internet connection. I give you that much. But it's actually interesting. If you go and see what's happening in Africa, they recognize this, and some of the most sophisticated science they're doing is computational biology, because they saw that they could leapfrog across some of the infrastructure issues. They're not very good with freezers, but they're really good with it's true. And so setting up a laboratory is hard, but setting up a bioinformatic network, it wasn't as hard. So, and I think that was facilitated by the fact that every consortium I named is putting up all the data free up on the internet. So I think that's one way to overcome it.
David?
One of the things that the medical system struggles with is the worried well. And I suspect if any one of us was sequenced, we'd find things in our genomes that we'd be confronted by. As sequencing becomes cheaper and cheaper, how's the health system going to deal with the worried well, basically?
Yeah, so you sound like a member of the U.S. Congress, to be honest with you. I probably just insulted you. I didn't mean it to be as insulting as it sounded. It's actually a common question I get asked. I mean, in terms of, while this all sounds great, but how are we going to afford it? You know, you could argue, how could you not afford it? I mean, I think, but that said, I think we as a genomics community have a responsibility. It probably speaks to that fifth domain of, we can't just throw this out and say, oh, well, this should just improve everything. We should have, as part of what we're doing, a research agenda that demonstrates that this can be more effective. You don't have to give, if we can better stratify cancer patients to get them the right medication, we could better do pharmacogenomics. In the long run, we should be saving money. There's a huge amount of waste just in adverse drug responses, and there's too many times we're giving people the wrong thing, or making mixed diagnoses. But I think we need evidence base to prove the point.
Can I draw you out a little further
Sure.
on that? So, you know, perhaps a technology that hasn't flowed easily to society has been genetically modified foods. How does a country or a system avoid what I would call the genetic disinformation that comes with adoption of new technologies like that?
Yeah, well, I may sound like a broken record, but one of the first things you do is you don't ignore the problem and you actually study it. So it's one of the reasons why we have always in the U.S. done this ethical, legal, social implications research to try to anticipate, you know, and even taking it, I mean, I'm just being half humorous, but even taking the topic around newborn screening and newborn sequencing, you can instantly engage in lots of reactions of people who want to really take it. So I think what you have to do is recognize what you're up against and start to enter into a discourse and to try to find out where is the comfort zone and where's
that battle has been fought very differently in Europe versus the US versus Australia. So do you see greater challenges in Europe around, for lots of historical reasons as well, around the idea that genetics and genomics is going to become mainstream in healthcare?
Yeah, so I don't know.
I realise you may not be responsible for that side.
Well, on the other hand, I might look at Europe, or I might look at Australia and wonder if your healthcare systems are better situated for these kinds of new technologies compared to the ridiculously broken system we have in America right now. So if anything, I wonder if, you know, there may be better places to pilot this than the United States.
I think we'll do 3 questions.
Okay.
In the middle, Suzanne, and then to the front.
I feel like we've been hearing a lot recently about the human microbiome.
Yeah.
Gut bacteria and so on. Do you see that as a big part of the discussion?
So I think it's a great question. Just really quick, the human microbiome is the collection of microbes that live in us and on us. Microbes outnumber us 10 to 1 in terms of cell number, so you're actually a very minority part of your little ecosystem. Just get used to it. And remarkably, 90% of those microbes have never been cultured. So we only know about 10% of the microbes. We are blind to the other 90%, and if we think for an instant that they're not relevant to health and disease, we're being naive. They're probably incredibly important. The sequencing technologies, these cool next-gen sequencing technologies, provide the ability. They're so sensitive, we can inventory those microbes just by sequencing samples, just purifying DNA, scrape a little bit of skin, get a little sample from wherever, and you can then inventory. Even if you can't culture the bug, you can find it by DNA sequence, and you can inventory it. That's led to major microbiome projects around the world that are learning a lot about the microbiome in health and disease. Where do I put it into the scheme? The microbiome is basically one of the most intimate environmental exposures that we have. It's really environment, but it's intimate because it's sort of part of our little ecosystem. And now we have an incredibly powerful tool of DNA sequencing to actually monitor that environment. You know, we probably can't monitor for half the chemicals we're exposed to, but we can now monitor for microbiome. And so you will see, just like what's happened in GWAS the last 5 years, I predict the next 5 years we'll see a huge surge in the number of microbiome papers. And I predict you will find there will be lots of things where we learn about the role the microbiome plays in health and disease.
Eric, first of all, thank you very much for a stunning talk.
Thank you.
And a comment before the question. I just, sitting at this point in time, appreciate so much what the US has done for science the world over, and speaking here as an Australian, what would we have done without the National Library? What would you have done without the Open Genome Sequencing Project? So great appreciation to the US.
Well, thank you. You just made my flight over here worthwhile.
My question is, we've been hearing about the bottleneck in bioinformatics from the very beginning of genomics.
Yeah.
So I wanted to explore with you in more depth what the US is thinking about in terms of training up the appropriate people. Are you succeeding? What should we be doing here?
Yeah, so let me actually give you a very direct answer if you really want. So we are, the NIH director, Francis Collins, just put a working group together of external experts that would advise his advisory committee, his highest level. And they worked for 9 months and they just delivered a report. So actually, if you want to read this report, if you just Googled NIH Advisory Committee to the Director, Data Integration Working Group, I think was the name of it, you would get a report. And you could read there what the recommendations are. Those recommendations are sitting on this laptop along with because now I've been asked to really play a major role in leading an implementation effort on campus. So I don't know the answers. And of course, all this there's some really grandiose ideas. Of course, that requires money, and money's tight and all that. But what I would say is I'm really confident that over the next year or two, we will be launching multifaceted attack on the big data problem. And I don't say for a minute I don't guarantee what we're doing is right. We're just going to do some grand things and hope that some of them work. Some of it will be training, a training aspect of it. Some of it will be making centers that create better software for everybody to use, not just the genome experts. And by the way, it's not just all about genomic data, it's imaging data, it's medical records data. And a lot of studies on should we be building big capacity at research institutions for housing all this data, or should we be putting it all up in the cloud, and all the things I'm sure many of you are thinking about as well. We just need far more research to look into this.
Last question.
Sure.
Glass half full and glass half empty. The genome-wide association studies have been kind of a 90% glass half empty in terms of finding the
A lot of missing heritability, right.
And given that the direct-to-consumer genetic tests are based on that data, what is your opinion on these direct?
So direct-to-consumer genetic testing obviously makes me nervous from the point of view of what it is based. Is incomplete information. It's very preliminary information. And I know a lot of really smart healthcare professionals who don't know how to deal with that data, and yet you put it in the hands of consumers. That said, it has created a little bit of a buzz that out there in terms of people getting interested in this, and that's sort of pushed the dialogue that I think in some ways has been interesting to watch. And I'm not going to be highly opinionated about whether it's a bad thing or a good thing. It's one of we live in a free world. That's certainly the United States, and people set up companies all the time. Actually, if you do a Google search, if you want to do something fun, do a Google search under genetic testing or genomic testing. And the direct-to-consumer ones aren't the ones that are nearly as wild as the, find your perfect mate through genomic analysis, find your perfect cosmetic, find your perfect food. I mean, they have essentially zero scientific validity as far as I can tell. And at least these direct-to-consumer genetic tests around complex diseases are at least using the literature to at least guide them. But it's premature. I mean, but it's been interesting to watch. And but at the end of the day, I want healthcare professionals in the middle of this because it's really going to be hard. And one more thing I really do worry about is I just don't want to have people buy into that, and then when it doesn't yield information that's useful for them, they say, you see, those genomics folks, they were lying the whole time. Because the point was, we're not there yet. And so, and I don't want people to be given a false impression that we are.
Just let me echo what Suzanne said and say thank you. We've had lots of lectures here. I think I've been at lectures at the Institute for the last 25 years. There are a handful that stand out. Eric Lander, David Baltimore, Harold Varmus coming through. I think you've given us a wonderful grand tour of genomics. I think you've done us a great service. I think we'll leave the auditorium inspired and optimistic about what, as a as a campus and individually, we can contribute to what's a wonderful effort. So Eric, thank you very much.
Thank you.